Morning. We're going to go over lecture. We'll start chapter 15 prior to the quiz. Okay? Chapter 15 builds upon what we talked about in chapter 14. Okay? So the idea of gene expression, transcription and translation as the process by which genes, the codes that everything that's coded in the DNA is expressed. All right? Basically, there are 4 different places, where, especially in eukaryotic cells, all right, where genes can be regulated. Okay? And I'm going to give you this handout. So there's 4 basic types of regulation. All right? I want you to keep this because we're going to go over some examples of regulation. All right? And as we go through the examples, I want you to sort of put them in different categories. Okay? You kind of have an idea of what's what. You know, one, you wake often? Yeah. Oh, you need this one? Stole it from you. All right. So, you can see on the handout, there's 4 different places where genes can be regulated. All right? At the transcriptional level, which means what? The level of transcription. So it's involved, what is transcription? mRNA. All right, so the production of an mRNA. All right, anything that regulates the production of an mRNA prior to it becoming a transcript. Okay? So transcription factors binding to a promoter, like stuff like that, is transcriptional regulation. All right? The next category is post-transcriptional. Where do you think that regulation would occur? Abby? Before it leaves the nucleus, but after a transcript is produced. All right? So RNA processing, right? The poly A tail, the 5' cap, export to the nucleus are examples. Where is this likely not present? In bacteria, right? Because we're talking about a step between transcription and before it leaves the nucleus that's probably not present in bacteria. So these are sort of the 4 categories for eukaryotic cells. Translational. Where do you think that regulation is going to occur? All right, likely in the cytoplasm, and how? Give me an example or describe how you think that will work. How a cell will regulate gene expression at the translational level. Abby? All right, so what about ribosomal binding? Right, ribosomes have to bind to an mRNA, right? Have to find the mRNA and then bind to it and produce a protein. All right, so that's the initiation of that translation. The translational complex is a level of translational regulation. And then finally, you have post-translational. So now a polypeptide has been produced. How can a cell regulate the function of the protein after a peptide is produced? And there's a lot of different ways of altering the function of a protein by modifying the amino acid residues on the protein itself. All right? So those are the 4 examples. As we go through this, sort of have them in the back of your head, have an idea of what they mean, and be able to sort of put what we talk about into those categories. Okay? Because on your exam, I'm going to give you an example, and I'm going to ask you, is it post-translational, post-transcriptional? What level of gene regulation would this fit into? Okay? All right. What's the important, why do we study differential expression, or what we call differential gene expression, or differential expression of genes? All right, or gene regulation. Basically, this drives everything that you've become. All right? Development, when you start as a one-cell organism, you have the potential to become this multicellular, multifunctional organism. And the expression of genes along development, when genes are expressed at the beginning as opposed to the end of a cellular life, or one gene being expressed in one area as opposed to another, gives cells their different properties, their different functions. All right, so it's very, very important. All right, so we're going to study how cells, prokaryotes and eukaryotes, alter their gene expression and what it means for the organism in general. All right, prokaryotes are unicellular organisms, though. They don't develop. They don't differentiate genes and become different cells. It is what it is. All right, but a prokaryote, a single cellular organism, has to respond to the environment. Okay? And prokaryotes regulate their gene expression in response to stimulus from the environment. All right? And so, two examples why we study gene expression. So prokaryotes respond to their environment, example being antibiotic resistance. All right? How many of you have heard of MRSA or MRSA? Right? Methicillin-resistant Staph aureus. Okay? That is a gene in and of itself, in the simplest form, is a differential gene expression. Staph aureus is a normal bacteria of your skin. Okay? In some cases, there are infections that develop, and the bacteria expresses a new gene against methicillin, which gives the bacteria a way of resisting a normal antibiotic. And that is simply a function of a new gene is being expressed in that bacteria in response to the environment around it. It's been exposed to methicillin in the past, and one or a group of cells has evaded death or somehow had a growth advantage, and now it produces a methicillin resistance. Okay? We went over the multicellular eukaryotes, and basically gene expression is most often regulated at the transcriptional stage. All right? Turning genes on, turning them off. All right? That's the simplest and sort of the most common way of changing gene expression. But there are other stages that are important as well. Okay? Questions up to there? Okay, let's try this. Oh, we went to 84. All right, so bacteria. They often respond to environmental change by regulating transcription. All right? So natural selection, and an example of natural selection would be the fact that bacteria have been exposed to different stimuli, and they have been selected over time for having a growth advantage, not maybe being resistant to certain antibiotics, whatever it might be. All right? So bacteria only produce the genes, they only express the genes that are absolutely necessary by the cell. So they live very minimalist lifestyles. All right? They don't waste energy in expressing genes that they're not going to utilize. All right? And we're going to talk about two examples of this in a second. All right? And a cell can regulate the production of enzymes. Really, so we're producing proteins, right? We're expressing genes, bacteria are expressing genes and producing proteins that are going to do something for them. Usually it's going to provide them some type of energy source. Okay? All right? So they're going to produce proteins and enzymes that are regulated by a process called feedback inhibition. What do you think feedback inhibition is? What's going to happen? Talk to your neighbor about it. I'll come up with some ideas, and then we'll share them around. Go. You all right? Yeah. Sure? I'll try that. Go to the elbow. There you go.
Last time I checked, you didn't, but that's like two.
I shrunk my sweater and washed.
Oh. All right, what do you have for ideas about feedback inhibition? What do we have? Anything? You didn't talk about feedback inhibition when you were just chatting? What do you think is going to happen? What is feedback?
So when they get energy back and they put it in.
In general, let's just say, what is feedback? If I ask you a question, if I want, so in a couple of weeks, I'm going to fill out CR2s and you're going to give me feedback about my teaching, right? And it's all going to be fantastic, right? What is that? So you're giving information about the state of stuff, right? You're going to give me information back. Okay? What is inhibition? To stop something. So the idea is, in some cases, a bacteria is going to produce, so here's the gene, right? So YFG, your favorite gene. This gene is going to produce a protein, all right, some globular protein, and in some cases, this protein is going to feed back and tell the cell that there's enough of this gene being expressed. Okay? So now it's going to feed back and say, and tell them what the state of the cell is. Does this cell need more of this gene? Does it not need more of this gene? Okay? In the case of feedback inhibition, what is going to happen?
It's going to tell it to stop making.
It's going to tell it to stop making. There's enough. We're okay. All right? We don't need any more. Okay? So this is sort of a very basic model of examples of prokaryotic feedback inhibition, gene expression. All right? And gene expression in bacteria, in this case, is controlled by a mechanism described as what we call the Opron model. All right? Genes in bacteria are sort of clumped together in terms of, we've talked about metabolic pathways, right? What's a metabolic pathway? Where you start with one precursor protein or substance, and it's broken down into, three other into another substance through a stepwise process. And each step is catalyzed by an enzyme. Okay? We talked about that when we talked about the beginnings of genes. Okay? The way bacteria sort of have structured their genome is that each one of those proteins or those enzymes, the genes for each one of those enzymes that control that metabolic pathway, are lumped together. So they're all expressed at one point. All right? This is what's called the Opron model. So you have basically it's a cluster of genes that are involved in one pathway. Okay? So here's an example of feedback inhibition and the operon model. All right? We're going to talk about two different operons, the trip operon, tryptophan, okay, and the lac operon, lactose. All right? So what you see here, here's your metabolic pathway, right? Here's your precursor protein. Enzyme 1 catalyzes to substance. The first step, enzyme 2, enzyme 3, eventually you end up with the product being tryptophan. Okay? So bacteria can synthesize this amino acid from a precursor protein or a precursor substance. All right? In order to do so, they have to produce these enzymes. All right? So here, when this precursor is present, this operon is expressed. Right? So there's gene for TRPE, D, C, B, and A. Those genes are turned on. Now what happens? What happens in the cell? This precursor protein is produced. These genes are turned on. Now those enzymes are available to produce tryptophan. Okay? So it will break down the precursor molecule and produce tryptophan. All right. Now, tryptophan can feed back. Here's your feedback inhibition here. Right? Regulation of the product of the pathway determines the activity of the gene. That's feedback. Okay? So now, feedback inhibition, product accumulates, it binds to the first enzyme, shuts it off. Because you don't want to have too much of tryptophan. Right? Product accumulates, it feedbacks to the enzyme 2, shuts it off. Until a time where tryptophan is no longer present, and then those genes that inhibition goes away. All right? So feedback inhibition. We're going to go over this in a little more detail in a second. All right, so the basic concept of operons. Right? They are a group of functionally regulated genes, and they are coordinated, they're controlled coordinatedly, or coordinately controlled. All right? Basically, it's a on-off switch. The precursor protein is present, operon is on. Okay? That's at least for the tryptophan operon, right? The regulatory switch is a segment of DNA called an operator. You'll see this in a second. Usually positioned within the promoter. Where's the promoter? All right, so upstream of the transcription of the genes that were being activated, right? And the promoter is a place where transcription begins. So this, like, binding of an operator, binding of a protein to the operator, and you'll see what this is in a little bit, is an example of a transcriptional control. All right? And so an operon is the entire stretch of DNA that includes the operator, the promoter, and the genes that they control. All right? You could almost think about the operator, that sequence of DNA, as being analogous to the Tata box in eukaryotic promoters. Okay? Questions on that? So, we're basically talking about a type of operon called a repressible operon. All right? So the operon can be switched off by a protein called a repressor. Repression is to shut something off. Right? So this repressor protein will bind the operon in some place, and what's it going to do? If a repressor binds an operon, it does what its name implies, which is stop what? Stop transcription. All right, it's going to stop transcription. All right, so it prevents gene transcription by binding to the operator and blocking RNA polymerase from binding. This is a transcriptional control. Okay, so repressor binding, it binds the operator and prevents transcription from occurring. All right, and the repressor is the product of a separate regulatory gene. Okay, so you have an operon, which is, for this pathway, has its operator, it has a promoter, it has all the genes that are regulated by it. Okay, fine. The repressor is outside of the operon. It is another gene entirely. It is not under the same control as the operator or the genes within that operon. Okay? Why is that important? What would happen if the repressor protein was part of the operon itself? Right? As soon as it got turned on, it would produce the repressor protein and then shut it right off. Right? So what does that do? That's not going to help the cell at all, right? Because we need the bacteria need those genes to be produced and be transcriptionally active in order to break down that or produce that metabolic pathway. So outside of this, you have the repressor protein, which can control that operon, right? And they separated the two. All right. Now, the repressor. We're getting into this is where it starts to get confusing. A repressor protein can be active or inactive. Right, in its normal state. So if a repressor protein is active, it's going to bind the operator. Okay? So in that case, an operon is normally in order for an operon to be transcribed, what has to happen? If a repressor is active. Repressor active, it's going to bind the operator. And transcription will happen, yes or no? No. Okay. So repressor active, operators bound, no transcription. In order to turn those genes on, what has to happen to the repressor? Has to be taken off. Okay. So in that case, and when a repressor is active, in order to turn the genes on, you have to inactivate the repressor. Okay. Opposite. When a repressor is inactive, what are happening to the genes? So, repressor is not active, genes are active. They're on. All right? So in order to shut off the genes, we have to activate the repressor, right? And then activate the repressor, and then it shuts off the genes themselves. All right? So, depending upon the state of the repressor, okay, and whether it's bound to the operator sequence, determines whether or not those genes are on or off. Okay? All right, so, and this is your example, the tryptophan, right? And a co-repressor is a way of turning on the repressor, right? So you have a repressor in an inactive form. Now there has to be a way to change it into an active form, right? So that you can stop transcription of genes. In order to do so, there's a binding of a partner, right? A co-repressor binds to the repressor and now changes it into an active form, and it can bind to the operon and shut it off. Right? Example being the tripped operon. Okay? So, in bacteria cells, normally, okay, the trip operon is on. Which means what? Is the repressor bound? No. Repressor stops transcription. So if transcription is on, repressor is not bound. Okay? And therefore the genes for tryptophan synthesis are on. Okay, fine. As tryptophan goes up, tryptophan actually functions the product functions as a co-repressor. All right? So, the tryptophan binds to the TRP repressor protein, and that turns the operon off. Okay? So the product of the pathway feeds back to the overall gene transcription by regulating transcription itself, by binding to the inactive repressor and turning it on. Shuts off the gene. Right? So the repressor is active only in the presence of its co-repressor, tryptophan. Right? So thus, the operon is turned off if tryptophan levels are high. What does it look like? Like this. So, top panel. Tryptophan is absent. Okay? So the co-repressor is not there. So your repressor in red is inactive. Okay? It's being produced by a separate gene. Okay? So here's your operator sequence. Right, here's your operator sequence. This is your TRP repressor producing the transcript, producing an inactive repressor. Right? This part is regulated completely differently from the operon itself. Okay? Has its own promoter, its own control. Okay? Here's your operon. Tryptophan is present. The operator is not bound. Okay? Operator's not bound. RNA polymerase can bind, produce a transcript, and as produce a transcript for all of the regulatory proteins, those genes are produced, and the pathway ensues. Okay? Those genes do their function. They then begin to produce what? They produce the product, tryptophan. Okay? Now, tryptophan increases in concentration. As tryptophan increases, now tryptophan is also the co-repressor. So now you move from here to the bottom panel. Tryptophan here binds to the co-repressor. So, tryptophan as a co-repressor binds to the repressor protein. That binding causes a change in shape, and the repressor is now able to bind the operator. Okay? So here now you have an active repressor. It binds to the operator sequence, and what occurs? Transcription stops. Emily.
Did you just say that tryptophan binds to the co-repressor, or is the
Tryptophan is the co-repressor and binds to the repressor protein. Okay? Sarah.
So this shutting off of transcription, is this like one of the examples that you were talking about of like 4 basic types of regulation?
You tell me.
Feel like it is?
You feel like it is. What does it fit into? Transcriptional control. Okay? So this is an example of transcriptional control. You're regulating the production of the mRNA. All right? So normally it's on, but now we're going to shut it off.
So what do you call it? Is it just like tryptophan?
This is, so we would say the TRP operon. Regulation of the TRP operon. What type of, so what are we doing here? Normally this is on, right? And we're shutting it off. So are we positively or negatively regulating this gene expression? Negatively. Okay? We're going to shut it off. This is an example of negative regulation. All right, so I gave you sort of this places, types of regulation where this occurs. But basically there's 2 ways that you can regulate gene expression. You can either turn it on or turn it off, right? Yes? You turn it on, that's positive regulation. You turn it off or lower the expression, it's negative regulation. Okay? And these 4 types I gave you on that worksheet or that note sheet are the ways that positive or negative regulation occur. Okay? Questions on the TRP operon? All right? So this is basically, there are two types of operons, repressible or inducible. Okay? A repressible operon is normally on and can be shut off, such as the trip operon. Okay? So the trip operon is a repressible operon. On the opposite side, an inducible operon, right, is one that is usually off. Okay? It's usually off, which means what to you? If it has a repressor protein, what is it going to be? If it's usually off, what's the normal state of that operon? Ben? It's not producing the genes. Okay? So what about the state of the operator of that normally off operon? So that normally bound, or that operon that's inducible is normally off. So what does the operator, what's going to be present at the operator? The repressor protein. Okay? So in this case, in an inducible operon, right, you'll have sort of an operator, right, and then you have the regulatory genes that are important for it. So let's just say A, B, C, D, E, right? Those genes. This being the operator. In an inducible state, the repressor protein is bound to that operator. So what does a cell need to do in order to turn these genes on? Somehow do something to remove the repressor protein. You have to inactivate the repressor. Okay? In order to induce expression of this gene, of these genes. So this is a form of an inducible operon, or inducible gene.